So let's start with Aquificae. You have a question?
Do we have to know the taxonomy?
No. So this is my first point. You're beating me to the punch. I do not expect you to memorize these trees, nor know all of this taxonomy. That's there for your own interest. I'm hoping that I mean, you probably have never heard of any of these creatures, but if you have, it'll ring a bell. And I'll do that for all of these groups we talk about, so that if you think, well, you know, I worked on Clostridium before, there it is, now I know what it is. I'm hoping to tie your previous knowledge to what we talk about. So that's the reason for this. The other reason I show these is because it gives you a sense of the scope of the diversity of the organisms. In the case of Aquificae, this is it. This is every genus in the file. Most of these are single species or a couple of species, and that's all. And so there's something like 24 species of Aquificae that have been characterized and named, and there aren't any others. That's all there is. So this is a very small, not very familiar branch of creatures. We're going to talk about Aquificae, and we're going to talk about Thermocrinus. Metabolically and physiologically, these organisms are homogeneous. These things are typically rods or filaments. They are all thermophilic, some extremely so, and they are all hydrogen oxidizers. Can you think of a chemical reaction that generates more energy than hydrogen oxidation? I mean, hydrogen is the definition of reducing power, right? I mean, quite literally, that's the measure that people use to measure reducing power. And oxygen is the definition of oxidizing power. And so they're burning hydrogen, not burning with a flame, but chemically reacting them to make water for a living. This is called the null gas reaction. Most of them are microaerophilic. This makes a lot of sense based on the fact that they live at high temperatures. The solubility of oxygen at high temperatures is really, really low. And so they're not used to being exposed to very high levels of oxygen. But they do need enough to run this reaction. They can most often use other sources of reducing power other than hydrogen, things like thiosulfate or sulfide, primarily. Typically not nitrate, so they're not going to be anaerobic. These things are autotrophs, so they get their energy from an inorganic chemical reaction, and they fix their own carbon. They do this using something called the reverse TCA cycle or the reductive TCA cycle. Have any of you guys ever heard of this before? So it's just, instead of running the TCA cycle in the normal route where you burn off carbon as CO2, you run it backwards and suck CO2 in and make acetate. That takes energy, of course, but these guys have plenty of energy from hydrogen oxidation. Again, rod-shaped or filamentous, they have an outer membrane. They very often have, you don't see it in here, but there's some, outside the outer membrane, there is a semi-crystalline protein array. So they have surface proteins studded all over the surface in a very organized way called an S layer. Most of them are motile and flagellated. If you look up the definitions, they're usually described as non-motile. And the reason for this is the way motility is usually measured is you put a drop on a slide, you look at it under a microscope, right? Imagine an organism that grows optimally at 85 degrees centigrade sitting on a slide at 20 degrees. How motile do you expect it to be? How motile are you when it's 65 degrees below zero outside? So if you heat the slides up, they swim around nicely. Many of them produce carotenoid pigments. The function of these is a little unclear. It may have something to do with oxygen sensitivity, resistance to oxidizing. And so they appear blue or pink, that sort of thing in the environment. Quite common in high temperature geothermal springs, hot springs, and underwater hydrothermal vents, this sort of thing. And so here's Aquificae pyrophilus, the first of these to be characterized extensively. Notice it's got polar flagella. Grows optimally at 85 degrees, and it grows pretty nicely at 95 as well. That's pretty spectacular. If you've ever picked up a glass bottle at 95 degrees, you're really impressed. This one species is the one that's able to use nitrate as an internal electron acceptor. So this can grow anaerobically by nitrate reduction. It was isolated from the Kolbensee Ridge, north of Iceland. You guys know that Iceland is basically an outcropping of the Mid-Atlantic Ridge, right? That's why it's so volcanically active, because it's a spreading center where two plates of the crust are pulling apart. There's some other members of this group, Caulderobacter and Hydrogenobacter, that have been studied before, but nobody knew they were special. One we've looked at is Thermocrinus ruber. And so I showed you these slides of octopus spring and showed you some pink filamentous growth in here. And the pink filamentous, the band of pink, all this is pink here. The reason for that pink color is the growth of this filamentous biofilm on those surfaces. And the primary organism in those maps is Thermocrinus ruber. We're going to talk later about how that organism was characterized and isolated. In culture, it grows as rod-shaped cells and they're motile. But in the environment, they typically grow as filaments. Thermocrinus ruber can use formate and formamid, growing heterotrophically, but that's not its primary role of growth method as far as we can tell. It is an obligate microaerophile. And again, this kind of claim to fame, it was attempts to grow this organism earlier on in the 60s and 70s that resulted in the cultivation of Thermus aquaticus, which in turn was the source of a whole bunch of interest in thermophilic enzymes for a variety of purposes, including PCR.